Good afternoon. This is my second try at making you some review notes to go along with this and talk you through the notes a little bit. Now, remember, you can type on yours that you've got on your computer. You've got, or you could print those out, whichever way you want to do it. So, number one, this is on the cells review page. These were all on Canvas. That's my dog's tail. Move, please. Nobody wants to see your butt. Why are cells so small? Explain the relationship of surface area to volume. Well, high surface area to volume, that ratio, if you've got super small cells, they're going to have a high surface area to volume ratio, and that lets more food in and more waste out. And that's good for the cell. And we're going to see that tomorrow in a lab. I'm going to cut up a potato and show that to you. Number two, nerve cells are really long and thin, and some reach all the way from your spine to your toes. And the lining of your stomach is rippled and folded back and forth like an accordion. Both of those, they are shaped. Their structure is going to help their function. So that long, skinny nerve cell is then going to have like these finger things that come out. They can touch cells that are very far away and take that message back to your spine. So they're able to do more work like that. The inside of your stomach, if you remember dissecting the frog, and if you took out the J-shaped stomach, when you slice it open, you can see it looks like this inside. And that allows it to actually open up more when you ingest food. And all that surface area now, it provides an area where digestion can occur, and you can actually absorb the nutrients from your food. All right, so more work can be done when you have increased surface area. 3, what's the function of the ribosomes? Excuse me, that's my dog. Go, please. The function of the ribosomes, it's the place where protein is made. Number 4, the endoplasmic reticulum makes up more than half of the total membrane system in many eukaryotic cells, so cells that have a true nucleus. Explain the difference between the rough ER and smooth ER. Well, rough ER is called rough because it's covered with ribosomes. So ribosomes make protein. Protein is made on rough ER, on the ribosomes of the rough ER. The rough ER can pinch off and make storage containers. And then it uses those containers to transport materials throughout the cell and out of the cell. Smooth ER doesn't have any ribosomes, so protein is not made there. The smooth ER is responsible for detoxifying the cell, so taking toxins out, and it helps to make fats, and it packages up proteins to be distributed. You might see these like black marks over here. When I write a quiz, I go through something like this, and every time I include a question about something, I check it off. That's how I know I covered all this material. So obviously I covered all this stuff on the quiz. Drug abuse damages what organelles? Well, drugs are considered toxins, and that means that they would damage the smooth ER because that's a detoxifier. The rough ER is covered with ribosomes. As proteins, they are made on the ribosomes. They get moved through that ribbon-like rough ER. Some of the proteins are going to have carbohydrates attached to them, so they're called glycoproteins. Glyco means sugar, so there's sugar protein molecules. I'm sorry, keep moving. Knowing the functions of the rough ER, what will probably happen next? Well, they're not going to make protein, but they could be stored in some of those compartments and then transported out of the cell. Number 7. Besides packaging proteins into transport containers, what are some other major functions of the rough ER? Some major functions then are also going to be making the storage compartments, and it's a place where ribosomes can make their proteins. Now, I keep repeating some of the same information in different ways because questions can be written in different ways. You have to be able to process the information and not just memorize. Like, I don't want to just give you a chart and have you memorize. I want you to be able to process that information into questions. Number 8. The pH inside a lysosome is 4.8. The rest of the cell is 7.2. that we know anything under a 7, smaller than 7, is going to be an acid. So we know the lysosome is acidic. What does this tell us about the function then of the lysosome? Well, our stomach is also acidic. Hydrochloric acid is about a 2. And if our stomach digests food, and because it's acidic, then lysosomes probably do some digestion. And they actually do. They digest old cell parts that no longer work. The breakdown or recycling of cell parts is called autophagy. Auto means self, and phage means to eat. Where does this occur in eukaryotic cells? It occurs in the lysosomes. Because they are acidic and they can digest things. Our white blood cells have a lot of lysosomes because white blood cells are a part of our immune system, and they go around and eat up like invader particles because you don't want those to make you sick. Describe the function of a water vacuole in a plant. Well, I kind of draw it here. The water vacuole takes up most of the volume of a plant cell. And when it's full of water, it pushes on the cell walls and it allows that cell to remain rigid, almost like, think about like a Lego. And then they are able to stack and make very tall plants. So it holds water that keeps the cell rigid and helps the plant stand upright. What's the benefit of the internal membrane system in the mitochondria and the chloroplast? Well, the mitochondria has that back and forth, wavy internal membrane. And the chloroplast has stacks of membranes that make little sacs. Both of those are increasing the area, the surface area where work can be performed. And that's going to allow both of those organelles to do more things. Okay, page 2. What are the functions? Well, sugar's made in the chloroplast of plants, and it's broken down in the mitochondria of all cells. So all cells need energy, and they break down the sugar molecules and release ATP. And ATP is the usable type of energy that living organisms can actually use to function, to fuel all their functions. 13. What might be some benefits to plant cells being surrounded by cell walls? Well, that rigid cell wall is going to allow those plant cells to stack, and then the plant can stand upright. A rigid cell wall also protects plants when they're living through some pretty harsh conditions. Think about they're living through rain and sleet and snow and, everything. So it's going to be important to help that plant survive. How do we benefit from the fact that plants have cell walls? Well, their cell walls are made out of sugar. It's actually a very complex sugar. It's a polysaccharide and called cellulose. And we also call it fiber. And that fiber is necessary in our diets to help us poop. It's really important we get rid of solid waste materials because they become toxic in our bodies. And fiber will help to absorb water in your intestine, your large intestine, and then help to clear that poop out of your intestines. And so it's really important that you are eating plenty of plant material like fruits and vegetables, because we really need to make sure you're pooping regularly to get the toxins out. Which structures make plant cells unique and different from animal cells? Well, that's the cell wall, chloroplasts, water vacuole. All right, there's a disease that causes or prevents certain cells in the liver from detoxifying poisons. So what's probably lacking in those cells? Well, the smoothie R are the detoxifiers. So it's possible it's missing some smoothie R. If a disease causes a person to release too little energy from their food, so they break their food down, but it's just not enough energy, the body cells might have a decrease in the number of mitochondria, because those are the organelles where energy is released from your food. Which organelles produce and modify lipids or fats would be the smoothie R. If proteins are made on the ribosomes, where can that process take place in the cell? Well, there are ribosomes in the nucleolus, there are ribosomes on the roughy R, and there are ribosomes out through that jelly-like cytoplasm. So that means protein synthesis can occur in any of those places. Which storage molecules pinch off of the Golgi body? And you can see that, I drew it on there for you. They are vesicles, and what will happen if those vesicles join together and make a bigger unit, a bigger storage container, then that's called a vacuole. And if they contain acid, then that becomes a lysosome. All right. Cell review or cell size review. I am not going to give you a drawing to label. I am not going to point to things and go, okay, what is A, what is B on a quiz. I'm not going to do that because these drawings are too hard to read. But you know the differences between plant and animal cells. Okay, so that one above doesn't have a cell wall. It doesn't have a huge water vacuole. It doesn't have any chloroplasts. So it has to be an animal cell and not a plant cell. I just labeled for practicing labeling. If you looked at your documents on when they were JPEG, the cells would be purple. So which one of those two has more surface area? The one on the left, because it's bigger. Which one has more mitochondria? Well, in the drawing, they're the same because I just took one drawing and blew it up from the other one. In reality, though, the larger cell is going to need more mitochondria because it needs more energy. And that's going to add to its mass. It's going to have more of all of the organelles, and it's going to make that bigger cell weigh more. What are the consequences for a cell if the cell membrane isn't large enough to have adequate channels for bringing in nutrients and removing waste? If there aren't enough ways to bring in food and get rid of waste, that cell is going to starve, and toxins can build up and kill that cell. Comparing those two cells, which one has a larger volume? The bigger one has more volume. It's filled up with more stuff. Now, these two questions, 9 and 10, really tell you the benefits of being a small cell. If a glucose molecule enters a small cell, it's going to be better able to get that sugar to the mitochondria, and it'll happen faster than if it's a big cell. And when those mitochondria start breaking down sugars, they release carbon dioxide, and it is toxic for the cell, so it has to be removed. Well, a small cell can remove those toxins much faster than a large cell. All right, remember you've got your charts. I don't have check marks beside nucleus and nucleolus. I am not specifically asking you questions about them. All right, but I checked rough ER, smooth ER, ribosomes, mitochondria, vacuole, Golgi body, vesicles, lysosome, and chloroplast. So there will be questions about all of them. It's 15 multiple choice questions, and 3 of the questions are about cell size. So that means, let me see, I'm doing 2 things at once here. If they're about cell size, then that means you really need to study that portion, and we'll talk about it more in the lab, either tomorrow or Friday. All right, some people have been contacting me because they're worried about their grades, and I totally understand. But really, it's going to be up to you. You are going to have to step it up, and you are going to have to probably change some habits if you want to see an A, for instance, in the class. So for one thing, instead of just reading my notes, I think it's going to be essential that you write the notes out in your own handwriting. Nobody can just read those and memorize them. I'm like Sheldon on Big Bang Theory can, but most people I know can't do that. What you can also do is then practice reading those notes out loud and really talking about the notes to yourself. That's going to help. I know you guys picked up study guides. It's a paperback study guide you got, and it's got all the chapters in it. So you go to the chapter about cells. There are questions within the chapter and questions at the end, and you could practice some of those if they have to do with what I've talked about. And the correct answers are in the back of the book. So that could help you. You could go on Google. All you do is go up to the search bar and you write in Quizlet for AP Bio cells, and I promise you someone in this world has made a Quizlet. I promise you that, because there's Quizlets for everything on there. So look for it. I know you can find this stuff. You guys are much better at technology than I am. You see that every single day in class. So I know you can find things. Study any review pages I give you. When I give you something and I talk about it, then it must be important. I do not review stuff that I wouldn't test over. I do not keep talking about stuff that I wouldn't test over. My questions are written the way I speak, and I'm trying to use the same terminology, the same vocabulary. So really listen to that kind of stuff, and then really read along with. If I give you a video, watch it a couple times. God, I know you don't want to watch anything and hear me, but watch it a couple times, because maybe it was made to really help you. Rewrite any of the information into your own words. It's terrific if you can read the science stuff and you're like, oh, I read all of it. If you can't explain it and process it, then it's not going to do you any good. I know a lot of people don't want to make flashcards, but they can help you. Flashcards for this one, it could have been that each organelle have a flashcard and then talk about its structure and function on the other side. That could have helped you. Different kinds of graphic organizers. We might have a situation where you could make your own chart, like make a new copy of the chart and fill it in to quiz yourself. Maybe we'll have a chapter where you're comparing two things, and you could have a Venn diagram where you're comparing those and contrasting those. Draw out information. Some people have to see the information like as a drawing, and you could do that. Draw it out. Draw out the organelles and talk about the jobs that they do. You have to remember about this class. It is a college prep course. You are going to have to step up your game. This is not like just the regular courses. When I'm teaching it, I have to follow the curriculum and the directions of College Board. That's the company that produces all of the curriculum for AP, and it produces the SAT, ACT, and other standardized tests. I have to follow their directions. I have to do what they say, and I have to go through the material they tell me to hit. Our book has 56 chapters. I have to cover over 30. So I can't slow down. I cannot slow down. I don't go any slower in the classroom. And actually we cover more material in the classroom. So if we were in person, we would be doing something every day, doing more activities, building things, turning in things. And we're not doing that right now because it's so difficult. Now, the State Board of Ed told us that for each of your classes, you're supposed to be spending 5 hours a week for each course. And I only see you 2.5 hours. So that gives 2.5 hours that you're supposed to be working on AP on your own. And what you could use that for is review. You could be using that time to rewrite your notes, to read your notes out loud, to use some different techniques to really prepare for quizzes. You might actually have to do something you've never done before, which is spend time every single day going through things. I never had to study till I got to college. In high school, never had to study. In junior college, never had to study. Junior and senior year, I transferred to a 4-year college and got my bachelor's. It was so difficult for me because I had no study skills. So it was really, really hard for me. It was a shock to my system that I had to start actually studying, and I had never done that before. And it would be much easier for you to learn those techniques now. Don't wait till you're in college to learn those. So spend some time every single day looking at notes, watching a video, reading your notes out loud. Remember, for AP classes, you get a weighted grade. You have to do more work than you would do in a regular class. That's just the name of the game. Because weighted grade for us means if you earn a C, you get a B in your final transcript. If you earn a B, you get an A, and if you earn an A, you get 2 A's. You are not going to get an A in here for just showing up and being nice and taking notes. You have to work for it. You're going to have to up your game. You know, you really are going to have to do that, because to get 2 A's in your final transcript, you're going to have to earn that. I'm not I'm cheating you if I don't have higher expectations, okay? If I don't expect you to perform the work of a freshman level biology class in college, then I'm cheating you, because this is supposed to be the equivalent of that. And, I'm not doing my job. I could easily be like, oh no, I want you to like me. It's going to be easy. I want you to like me, but I can't make it easy for you. I can't do that, because then I'm not doing my job as a teacher. So I have to challenge you. You can do this. Everyone can pass the tests, the quizzes. You might not get an A, but I know you can pass it. You have your notes to use. For crying out loud, don't forget that. You have notes. And you can make your own notes. You can add as many note pages as you want. You're at home. How do I know? Another thing is, don't forget, there are videos to watch on YouTube. So if we were in person, I would take each week a few minutes out of a class to watch one of the videos. And I stop them, during that video, and we talk about things. So with any of these, go to YouTube and just type in the search bar, type in your topic. So this would be cells or cell organelles. Type it in, and Bozeman will come up, Crash Course, Amoeba Sisters, 18,000 more will show up. You pick the one you like and watch it. So maybe to review one day, you study your notes, and maybe another day you read your notes out loud, and maybe the third day you watch the video. Or maybe you're waiting for your mom to pick you up and take you, somewhere. So you got some time, hurry up and watch one of the videos. It's gonna be like 7 minutes. Watch one of the videos. You're gonna hear the same material, but you're gonna hear it in a new way. And that's gonna help it get ingrained in your brain. I know there was something else I wanted to tell you guys. I don't remember what it was. The other video, I know the sound was absolutely horrible. So I will probably erase that one and just add this one. Tomorrow, which is Thursday, 2, 3, and 4, we're going to go over the lab about cell size. If you have questions about the quiz, you've got to ask. You have to either email me, you have to put it in the chat during class, you have to turn on your microphone and ask. I am not a psychic, okay? I can't read your mind and know what your questions are. You have to ask me. If you have, and then on Friday, I'll see you guys. I'll put the quiz out probably at noon on Friday for everyone, and you'll have till midnight Sunday night. It's 15 questions. Remember, 3 of them are about cell size. The others are pretty much, each one of the organelles has 1 question apiece. And it's pretty much all about function for those. Now, if you've got techniques that seem to work for you and you are doing well, A's, B's, even C work. If you're doing that and you want to share it with other people, email, let me know what you're doing. And I'm going to put together a document that will have a lot of suggestions of different things you can try. And each time, try something different, okay? If this doesn't work for you, try something else. If writing out the notes doesn't seem to work for you, then try watching videos, or try talking about the notes, or drawing pictures. So try something else until you find what works for you. More than likely, you're going to have several different techniques, because different ones will work for different topics. Okay, so I will see you in the next couple days. The quiz does not come out until Friday at noon. You have everything you need. I go over these every single time, and I think some of you have realized it already. I tell you what to review. I'm not tricking you. I'm not trying to trick you, okay? I tell you exactly what to review. You just have to review it. And I can't give you the exact questions. I can't do that. So you start thinking, okay, what could a question about lysosomes look like? What could a question about the Golgi body look like? And start thinking in that, think backwards, make the question in your mind and then, come up with your answer. So, try to process that information, and that might help you. But again, if you have questions, please, you have to ask me. I can't read your mind. Okay? I'll see you soon. Bye.